already see your screen, you know, and then we start around 11. We give like 10 minutes for people to come in and join the room. Daniel, you're introducing, right?
Yep.
Fantastic. Awesome. So let's give a few more minutes. A few more minutes and we can start. Meanwhile, I'm going to make a cup of coffee. Nice meeting you, and thank you so much for speaking for us.
Thank you for inviting me.
So as I said, so Tarcísio works with zebrafinch, and Diego studies the rat vocalization, and Rodrigo too. Actually, you know, they both study rodent vocalization.
Oh, nice.
Yeah.
So using rats, mouse.
Rats. Mm-hmm.
Hi guys, good morning. I'm driving a car, but I'm plugged in. I'm very excited to attend to this talk. Welcome to our institute.
Thank you.
Well, hopefully next time you will be able to visit in physically, you know.
Well, maybe it's nice that this pandemic travel is very simple. Are you already back to lab, or you're still at home?
Yeah, I'm mostly at my home office. Yeah. But I think slowly we are trying to get back. But we are now moving to a new building, so we are not on the transition because of the pandemic, but also to the new building. So.
I see. I haven't timed my talk. I don't know how long it will go.
Yeah, it will be fine.
Do you have this seminar every week?
Yeah, every week. This seminar has been going on I think, consistently on Fridays this time. Yeah.
Because you can invite more people from other countries.
I think actually this seminar actually has been going, I think, for a long time, even before the pandemic. But I think now it's true. Now it's become much easier, right, to invite also. Yeah. But actually, yeah. Yeah, this online seminar, there's something funny that is going on, and so because I sometimes participate in some math seminars, and what I see is that the same person now is invited in several seminars, and they're giving exactly the same seminar. So it's kind of, there is a lot of overlap now.
Oh, yeah.
There has been a proliferation of seminars. In fact, like this is when we first closed down, we were the only ones that with our university actually doing online seminars and broadcasting. And then all of a sudden, in 2 or 3 months, there was this explosion of like, now everybody's doing it.
Yeah, there are like 3 seminars per week now.
Yeah.
And so which is kind of interesting, right? So there's something called video call fatigue, which apparently you get more tired from watching from different things, because you have no necessarily other cues other than the sound, very few visual cues, and even your own face on the screen distracts you, and it apparently makes you more tired. So I get that's all I do all day long. At the end of the day, I'm like, whoa, I can't do this anymore.
No, it's true. It's true that and also I feel that just because it's online, people think that it's much easier. So you have like a lot of meetings every day.
Yeah.
Like,
Yeah.
I remember this Tuesday, I was thinking I had like a 7 meeting. I said, okay, come on, I cannot do this anymore. It's true.
Yeah.
Wow.
Okay. Good morning.
Good morning. Yeah, we already have a few people waiting on YouTube. So it's 11 o'clock. So if you guys wanna kickstart, Daniel.
Yeah. So it's fine, easy. We start.
Yeah.
Okay. So So welcome everyone, and it is a great pleasure to have Dr. Isi Zhang presenting her recent work to us. And I just want to give a little bit of her background. So Isi is currently an associate research scholar at Princeton Neuroscience Institute. And she did her undergrad in physics at Tsinghua University in China. And then she did her PhD in Penn State, studying the neural-based vocal sequence generation in Bengal finch, right? And then she continued her studies during her postdoc on vocalization, but now in a different species that we know very well, which are the Marmosets. Right? So in Brazil we call saguis. And she's a specialist, so therefore in vocal communication, both in birds and the primates. Right? And I have known her for a while now, maybe 5-6 years now. Yeah, we've been working. We work in several projects together. And one thing that always amazed me was actually her depth of her ideas, and I think the new techniques that she introduced to the studies of vocal communication in primates. Therefore, I'm very curious to know what she will have to tell us today. So without further ado, I would like to welcome Isi Zhang. Thank you.
Thank you, Daniel, for the nice introduction. And thank you for inviting me to share with you some of my recent work. Maybe I should also thank the pandemic as well, as the physical travels now are really hard, but this makes it very easy. And maybe also because of this, we should realize that these studies in acoustic communication, vocal communication, is a very important study, as when physical travel is limited, animals and humans both use acoustic signals to communicate. And so, all right, so as Daniel introduced, I worked with both birds and marmosets. And so I know some of you guys also work with a number of other species, like rodents and maybe fish, on vocalization. And so today I'm very excited to talk with you guys to discuss the ideas, and also please feel free to ask questions. All right, can I start, or should I wait? All right. So today my topic is the cardiorespiratory basis of the hierarchical organization of vocal communication. So you probably all know that vocal communication is ubiquitous across the animal kingdom and also in humans. So from fish to primates, all these animals use somewhat similar yet different enough mechanisms to generate sound. So animals use vocal communication for different purposes. For example, in the birds, so they sing to attract the females and to protect their territory. And for example, the cats mostly meow to just humans. And the marmosets, you probably hear a lot of the marmosets in Brazil that they use these contact calls that are very loud and can propagate very far to make contacts made with other marmosets at a distance. So these marmosets are very nice because they share a number of similarities with humans, especially in their social structure, their breeding strategies, which are organized to get around with this problem of the extensive demand of the parental care when their babies are very young. So therefore, this vocalization plays a very crucial role as it helps the young marmosets to communicate with their parents when they need to be taken care of. So like human babies, these marmoset babies start vocalizing from the very beginning of their life. And from these pictures, you can tell that both infants, the baby, the human infant and the marmoset infant, they really spend a lot of effort on vocalizing. So in fact, for young infants, human infants, crying is the most energy costly activity among all other physical activities. Yes, we think that this vocal production is tightly linked to the regulation of energy. And that's why today I'm going to talk about these investigations about the relationship between vocal production and the cardiorespiratory dynamics. So for many species, it actually makes a lot of sense for the vocalization to be the first motor activity for them to be able to carry out. Because respiration is so vital for all animals to survive. And also this vocalization, it basically recruits this respiratory system. So it makes sense that when they are able to breathe, they might be able to vocalize as well. And the mechanisms to produce the sound among the mammals are very similar across different species. So they all use this larynx that is like a voice box, and it has a membrane structure called the vocal folds. And when you breathe, you push the vocal fold to vibrate, and that's the common mechanism for a lot of mammals, including marmosets and humans. So by coordinating these laryngeal activities and the respiratory activities, you can produce different kind of sounds. Okay. All right. So as these infant marmosets, once they started vocalizing, they can continuously produce a variety types of calls. And these calls are the result of this coordination between respiration and the laryngeal movements. And these calls are organized with some sort of structure. And if we zoom in this long sequence, we see that this kind of relatively stable structure with repetitive utterances of these tonal features. And these are the calls that you're very familiar with, which are the Phee calls. And these Phee calls are the marmosets' most energy costly, and they are very loud, and they are like a pure tone calls, and they are also the longest calls. Okay, so if you keep zooming, you see that there are these discrete syllable-like structures. And so the duration of each of these utterance, we say, should be determined by respiration. Okay? So on the right, we see that this is a Phee call from adult marmosets, and we took the EMG signals, which is the signals of their muscle activity, okay, of these cricothyroid muscles, which is the muscle that controls the stretch of the vocal fold, and also the abdominal muscles, which are the muscles that supplies the expiration movements. Okay, and so these muscles contract during the production of Phee call. And the duration of this muscle contraction actually corresponds to the duration of these utterances. And so if the duration of these utterances are limited by respiration, we should expect that these changes over development, as these marmosets get bigger, we should expect to see some change in this duration as well. So if you look at these kind of Phee calls that the marmosets produced from first week, fourth week, and seventh week after they were born, you see that the total duration of the Phee calls is not consistently changed. But for individual utterance, you see that they are becoming longer and longer. And in fact, this trend is quite linear. So development, as it changes the size of these marmosets, it changes basically the timing of these individual syllables through affecting the timing of respiration. So, but if you look at the developmental change of the marmoset vocalization, it's not only such linear kind of stretch. There is also a lot of nonlinear changes occurring across development. So if you notice that there is this kind of broadband calls that gradually disappear, and these calls, we will call them cries. They are immature. And these calls are not used in the adult marmosets anymore. There are also these short calls, like here. So these calls also disappear in this context. So this context is in when these infant marmosets are alone and they just spontaneously produce vocalizations. And these short calls, they change their usage over development. And in adults, the trills, for example, are used when the marmosets are closer with each other. So the question is then, can the changes, the kind of linear change in the body size also affect such kind of nonlinear change across development? So here showing is that over development, the proportion of Phee calls becomes greater, and it's getting closer to 1, whereas these other short calls like the trill and tweeter, they become more and more rare and decays to 0. So we want to test if this long growth can also affect such kind of change. And the way we tested it was by placing this marmoset in a helium oxygen environment where this air is much lighter than natural air. Okay, it's like 3 times lighter. So the idea is that when the marmosets breathe in this kind of air, because it's lighter, so it takes shorter time for them to breathe. Okay, and that simulates the effect of a smaller lung. And if that works, we should expect that this kind of timing change may or may not have some effect on this kind of nonlinear change of call types. Okay, and for this experiment, for day 1, we place them first in air, like normal air, for 10 minutes, and then place them in these heliox for another 10 minutes. And for the next day, we swap this order. Now we analyzed the data. We took only the last 5 minutes when the gas density is pretty much stabilized. And we first confirmed that in heliox, really, the marmoset breathe faster than in the normal air. Okay, and one effect is directly on the duration of these Phee utterances. They really produce shorter Phees in the heliox, which is what we predicted. But more interestingly is that in this heliox environment, these trill calls or the tweeter calls, which are the short calls, are also enhanced in proportion. So this really similar to the effect as if these marmosets were younger. So at least we think these changes in lung size can partially affect these usage of this call through development. Okay. Okay, so that was the analysis of the time scale when individual utterances are produced. Now, if we zoom out and take a look at this sequence of vocalization, so actually at longer time scales, these vocal sequences exhibits a change in the temporal structure as well. What I mean is that if you look at coarsely look at these vocal sequences by extracting their acoustic features, and we see that, for example, here we extracted the Wiener entropy of this sound, and we see that this entropy fluctuates slowly, and the time scale is on the 10-second time scale. Okay, and a high entropy, so entropy measures how noisy the sound is. So higher entropy are noisier sound, and the lower entropy are the tonal sounds. So this kind of alternation exhibit a frequency around 0.1 hertz here. So through development, this kind of oscillation also changes. So in the beginning, actually, the oscillation is quite noisy. Here it's plotted the acoustic power of this sound envelope. So we see in the beginning, it's quite noisy actually. But gradually, these kind of oscillation became more and more regular, like periodic oscillations. And if we plot the power spectral density, we see that the first week's peak is actually pretty broad, and the peak becomes more and more sharp as the marmoset grows older. So if we analyze this kind of oscillatory pattern, we see that in the in this orbit diagram, which is so if we plot this oscillation of this acoustic power in a phase space, on the x-axis is a time the acoustic power of time point t, and on the y axis is the time point with a fixed delay. And we see that in the beginning, it's very stochastic, but as it grows, it becomes more and more like a periodic oscillation. And we see that around after around like the 30th postnatal week of postnatal day, these kind of periodic oscillations became more dominant. So the question is like, what can push this kind of bifurcation like change that makes this emergent, makes the emergence of these periodic oscillations. So the hypothesis is that one possibility is the energy. So why I said is that, so for the acoustic features, for example, like the Wiener entropy or the fundamental frequency, to produce a Phee call, which is the symbolized like the maturation of these vocalizations, we see that it requires the stretch of the vocal fold to be to like very long. And to make a loud call, you need to provide a high pressure. So both loudness and the tonal kind of feature requires a very hard stretch of very hard muscle contractions. So both would cost more energy. So maybe in the beginning, because of the limit of energy, it is hard to produce this kind of Phee calls. And that's why they produce all types of vocalizations. And as they grow bigger, they are more energetic. And so they are capable of producing more of these mature sounding calls. And if that is the case, these energetic constraints might be one of the reasons that they change their vocal behavior. So we're gonna show that energy development actually can drive this kind of periodic oscillation. And so we're gonna take a look at the energy supply in real time as these marmosets are producing vocalizations. The way is by looking at the heart rate change during vocal production. So here showing the blue curves are the power, acoustic power, and the red ones are the heart rate change. So we see that during both earlier periods and later periods, this heart rate change seems to follow pretty closely with these acoustic change. And it's very obvious that from these later ones, there is always an increase of heart rate prior to the production of a call, and a sharp decrease upon the call production. And then heart rate starts increase again after producing a call, and then it fell again after the next call is produced. So if we calculate the coherence between these two signals, we find that there is a pronounced peak around 0.1 hertz for both earlier and the later calls. And the later calls has an even stronger coherence. So in fact, the metabolic energy increases during development. And we measure the heart rate and plot the heart rate against the body mass. So we estimate that metabolic energy is proportional to the amount of blood that's pumped by the heart per unit time, okay? And that would be determined by the size of the heart and also the heart rate, okay? And if we do that, we estimate the metabolic rate is approximately proportional to body mass to the exponent of 1.4. And then we look at the acoustic energy that's being emitted to the air per unit time. So if a fixed amount of energy is used in producing sound, we should expect that these kind of acoustic energy emitted per unit time also scales roughly with this metabolic rate. And we see that this energy rate scales with body mass also around 1.4. So this signal naturally is noisy, but statistically we see that these kind of exponents is not different from the predicted one. So through development, more energy is emitted to the air.
Sorry, I would like to ask you a question, if possible.
Do you have questions?
Yeah, can you hear me?
Oh, yes.
Sure. No, I'm really enjoying this. And I was actually wondering, before we move forward, because you were following this sort of as a continuous fluctuation, I'm thinking the different kinds of calls, the Phee and the trill, are they not distinct motor programs in larynx control and such?
So they are distinct larynx movements. Yes.
Okay, but so maybe let's see where this goes, because I'm thinking it seems to me like there is like in the whatever PhD or something, there is someone deciding, okay, now we're gonna do a trill, now we're gonna do a Phee.
Yeah, so I can show you after the talk that I have a model. Basically, the idea is that because of the nonlinear properties of these CPGs, the central pattern generator, essentially what we would expect is that if you have a linear change in the drive signal, that cause some nonlinear change in the CPG activity. And because of that, you see these kind of discrete-like changes in the pattern. Like sometimes it's like a Phee, which are long ones, but sometimes part of the CPG can oscillate faster and produce something like a trill or tweeter. So that's
fascinating, because like in the birds, there is, I mean, I follow the work from Gabriel Mindlin in Argentina, and I mean, there is a similar story, but the nonlinearities they express in the output, right? They get expressed in the actual, I mean, in the periphery, let's say, not in the CPG. Like, so you're saying, oh, that's interesting. Okay. So you're saying that these nonlinearities are going to affect the motor program in the
yes.
In the motor
so
loop.
actually, I read a lot of their work, and the idea I got is really from them. And they also have some papers about this kind of nonlinearity can happen in the brain, essentially. for example, they cool down these, I think it's canaries.
Yeah.
And what they saw is that these syllables, instead of linearly stretch, they also break into like shorter pieces. And this kind of nonlinear change cannot be explained by the peripheral, but has to be explained by changing the.
Right. Okay, now I see where you're heading. I'm gonna keep enjoying the thank you.
Mm-hmm. All right. So basically what I'm trying to show here is that through development, this metabolic energy increases, and it's reflected in their vocal production as well. So can we make a model to simulate this kind of effect? So through development, if we think that a fixed amount of energy is used in vocalizing, and if we don't change the context, we expect that this kind of allocation of energy is the same. And so we coin a work energy budget, which is how much energy this animal is invested in producing vocalization. And this action of vocal production is constrained by this energy budget. And so in this model, it's very simple. So there is a neural part that its activity directly drives the muscle contraction, which produces sound. So it's very simple. And the details of the sound doesn't matter. It's just the larger the muscle contraction is, you produce a louder sound. And the amplitude of the sound will provide a positive feedback to the drive. And it will keep this spontaneous vocalization going. But the positive auditory feedback cannot be like keep adding, because then it will just blow up. So now we have this energy constraint. So as the marmoset produces sound, it costs energy, and it reduces the energy available for the next moment. And this energy availability will scale the signal of the drive. Okay, and this drive basically would, if it's high, it will produce a sound, and it produces sounds. As it produces the sound, it drags down the energy, and which would drags down the drive. Okay, and besides, there is a noise that's fed to the drive as well. So it's a very parsimonious model. And one crucial part is that there are these time delays as neuroactivity to produce a sound, and also from the sound that will make a change to the drive, and also for the energy to make a change to the drive. So these delays are important. And if we simulate it, we see that there are two regions in this bifurcation diagram. So in the region 1, essentially we don't see oscillatory, these periodic oscillations, but in region 2, we see periodic oscillations. And these two regions are in the space of the energy budget and the alpha. And alpha is how strong this positive feedback is imposing on this drive. So basically the idea is very simple. So these sound, if the marmoset experiments with the vocal apparatus and make a sound, and it would make the marmoset to want to continue producing more sound. And it acts as a reward to producing louder sounds, okay? And so if this alpha increases as well, it actually can make the crossing happens earlier. So that is similar to like Daniel's findings that when these marmosets are provided with the contingent parental cause, it accelerates the vocal maturation. And I think here it basically implements that idea into this alpha. And when alpha is higher, the slope becomes steeper and actually can pushes the day of vocal maturation to earlier days. That simulates the effect of the acceleration. Okay. So here is the result of the simulation. So in the region 1, essentially we don't see very regular oscillations, but there are occasional ones, which are the oscillations when the system is pushed by noise across this boundary, and it will produce some loud sound. But then in between, it's mostly driven by the noise. But as it crosses the boundary, it faithfully produces kind of periodic like sounds. So using this very simple model, we can actually kind of simulate this kind of emergence of oscillations occurring in their vocal production. Okay. All right. So far I have talked about the findings of how respiration and energy can affect these vocal sequences at different time scales, and how it can change vocal patterns through development by affecting energy or the biomechanics. So now the question is that if vocalization is tightly related to this cardiorespiratory activity, if we think it in an even broader view, these vocal production may just intersect with the regulation of energy of the entire body. And that is regulated by this kind of homeostatic control of the brain. And if that's the case, we should expect that this vocal production shares some common brain areas or brain networks with this homeostatic control of energy or the respiratory or cardiorespiratory activities. Okay. So the hypothesis is the homeostatic control at rest and vocal communication shares common brain networks. So at resting state, the marmoset is free of external stimuli. So it's basically the brain activity reflects essentially only the internal dynamics. And so an alternative hypothesis is that during vocal communication, these distinct brain networks is recruited other than this kind of default mode like activities. So to test this, we used this functional ultrasound imaging technique that Daniel and Ahmed, they pioneered in behaving marmosets. So probably you have heard about it a lot. It's a new technique that measures the change of cerebral blood volume with a high temporal and spatial resolutions. And it can measure the brain activity of a large cross-section of the brain, including both cortical and subcortical areas. And so I'll quickly summarize what Daniel have found in this experiment. So basically Daniel find out that during vocal perception and production, not only the documented vocal network are responsive, but also the areas related to social behavior are responsive to this kind of social vocal cues. And this includes a lot of brain areas on this mid-sagittal section. And essentially, they include a vocal network, mainly the ACC, the anterior cingulate cortex, and also has these medial thalamus and the PAG. So I'll just point out those ones. And also the social behavior network that includes this lateral septum, or these ventromedial hypothalamus, and also PAG. And here are Daniel's results. Basically, he designed these playback experiment and measured these brain responses to those played marmoset vocalizations. And what he observed is that there is a strong response to marmoset calls in these anterior cingulate cortex and this lateral septum and some thalamus areas, and a little bit of this hypothalamus, I guess. And also for call production, some similar areas are responsive, and also there is the strong activity at this PAG. Oops. At this PAG. Okay. So the question is then, can we observe similar kind of networks being activated during resting state, during the resting state? So the way I analyze this to extract the instantaneous activity of the brain during resting state. Okay, so resting state is when the marmoset is just sitting there without being played any vocalizations, or there's no other animals in the room. So basically the animal is relaxed and sitting there by itself. And so essentially, I first reduce the dimensionality of these images by extracting the spatial principal components and calculate the outer product with wavelet packet transform of each pixel. And basically by doing that, I extract both spatial information and also the frequency temporal information from these signals. And I did this for 60 sessions. Each session lasts like 500 to 1,000 seconds. And then I further decreased the dimensionality by embedding that signal onto a 2D space. And this technique is called the T-distributed stochastic neighbor embedding, TISNE. And so now each frame becomes just one time point. Becomes just two points, okay? So the dimension has been much reduced. And this is just to help us to visualize better changes in the like some global change in the states. And if we plot these 2 dimensions, and we see that there are some clusters, and we can segment the clusters. And here, what happened? So here are the exemplars of some of the clusters, okay? So for example, this cluster, it's very strong. You see at low frequencies, that's weird. At low frequencies, you see that there is a strong reaction at the anterior cingulate cortex. And this is at the frequency of 0.03 hertz. So maybe I should explain a little better what this means. So this is that I extracted the points which are each each point, so this cluster are a bunch of point, and each point is from one time point, okay? And so for that time point, I get the wavelet packet transform of each pixel of the brain. And then I stack all these brains from that same, all these frames from that cluster and take them in. And for each frequency, okay? So then this is 0.03 hertz, and this is like 0.09 hertz, and so on. So let's just take a look at this. So for this cluster, you see this kind of pattern, and it's mainly within these slow frequency regions. And this cluster, you see strong activity at these cortical areas. I think it's the area 31. And also you see kind of strong subcortical areas at a high frequency. And for this cluster, basically you don't see much. It's more like a baseline state where there is very little activity actually in these areas. Okay, so those are the three exemplars. Now, if we take the first state and compare with this activation map that Daniel got from these cost perceptions, okay, and we see that actually a lot of areas that common to these vocal activation map are illuminated here, like these areas 32, area 14, 25, and also these social areas like the lateral septum, preoptic, and also these medial thalamus. And at a higher frequency, basically you see even stronger and consistent activities of these areas, and also including these ventromedial hypothalamus. So it looks like at some episodes during these resting states, there are some common patterns that there are like activities of these brain areas that share very common patterns as these vocal communication related areas. So next, if we, next question is then, do these resting state activity have anything to do with energy, like heart rate, for example? So here I plot heart rate together with that ongoing low dimensional brain activities. And we actually see that their activities follow each other pretty closely. And it might make sense to think as the signal we took are like blood flow, and the heart rate change will definitely be reflected in the blood flow in the brain. But in fact, if we do a causality analysis, we see that the causal relationship is not only from heart rate to brain, it's also from brain to heart rate. And this technique is by using this partial directed coherence that Daniel kind of invented. It's like a variation of it. And we see that from brain to heart rate, there is a very strong PDC around this 0.03 hertz, which is like that one we just observed. We just looked more closely. And so from heart rate to brain, basically the strong coherence occurs at some different region of frequencies. So together, these results tells us that this resting state activity shares common brain networks with vocal communication, and also they are very tightly related to heart rate change. Okay, so To conclude, so far I have talked about mainly 4 things. First, this cardiorespiratory activity affect the vocal production at different time scales. The second, changes in respiration energy can non-linearly alter vocal dynamics during development. And third, resting state brain activity overlaps with the social vocal network. And last, this brain state and heart rate fluctuation can causally influence each other at different frequencies. Okay. So that's the end. And I just want to take this opportunity to thank all the members in the lab, and especially people who have been involved in these projects. So Daniel and Ahmed, they have put a lot of efforts in this development of this fusi technique. And the Kuhn elements in Southern Denmark University, I did a project there to measure these biomechanical properties of the marmoset larynx. And then we have done a lot of experiments together. And also like John, Chago, Lauren, they're all involved in these projects. So, and thank you for listening. I would be glad to take questions.
Thank you, Phee. Yes, we're open for questions. Anybody that wants can speak directly, or if you prefer, I will transmit the question for Phee. Well, go ahead, Diego. I was ready.
Yeah, great. I really enjoyed this. I was wondering. So now you have all this model about fluctuations in maybe in energy consumptions and such, sort of being behind the transitions in vocal production across development and such, right? So when people approach vocal production from a purely, ethological perspective, people expect that the call selection is driven by social behavior needs. I don't know. So if the animal produces a Phee versus a trill, maybe the brain is implementing it's solving some social issue or something. So now you think that call selection is mostly driven by this energy fluctuations, or is there room for that being, that depending on behavioral input, social needs?
Yeah, so totally. So maybe I should have mentioned. So these experiments are on just this single type of context when this marmoset is by itself. And this is the context. And we had actually experiments before. Let me find that these marmosets use different calls in different contexts. So essentially, if you have a single marmoset, and it will just produce these Phee calls, and if you have two marmosets that are far from each other and they cannot see each other, they exchange vocalizations by these Phee calls. But if you remove the curtain between them, and they can see them see each other now, they produce more like these trill Phees, which is like a transition between Phee and trill. And if you move them even closer, they pretty much just produce these trills. And you see that there is a very consistent change of this type of call usage, depending on the context. So for my analysis, they are pretty much all at this context when they are alone, okay? And that corresponds when the moment that they spend most energy to vocalize. And then energy becomes a factor that matters to them. But as they become closer, then they don't spend as much energy to communicate. And then energy becomes a less important factor for them that limit this behavior.
Could it be that, I mean, some of these changes, depending on context, are actually affected by, I mean, it could be secondary to their behavior changing, and then their metabolism changing, and then their energy sources changing, and then this affecting call selection? I mean, could metabolism also play a role here in this graph?
So yeah, what I think, so I'm not sure if there is a definite answer yet. What I think is that this vocalization basically is like a sensing signal. And it senses the other marmosets, and so if they're far away, they make a very loud sound so that it can reach out to a distance that's far. And if they are close to each other, they don't need to make so much effort to make a loud sound, because there are other information, like they can see each other, they can smell each other. So there are a lot of information that supplement the information of where the other marmoset is, what it is doing, or if there is food around, and those kind of things. So they don't need to produce these sensing signal that hard. So I think these kind of environment setup actually have a very quick effect on how these marmosets would select what calls are used.
No, but I was thinking, like, maybe when they're when they're close together, maybe they're jumping around, they're doing like much more active behavior, and then that also like goes in the energy source and the energy availability.
Yeah, so I don't know how other, I'm not sure how other behaviors are like interact with this vocal communication system yet. So there, I don't know if like locomotion may have some effect on vocal production as well, and that would be interesting to understand. Like if they have to travel very fast, for example, of course, you can expect that they don't vocalize as much, probably.
Yeah. And then it's interesting because this is something that I think with the rats, right? I do similar things with the rats, and like even if, and I'm more on your side. Like I actually like this line of seeing how sort of like metabolic and behavioral state actually constrain a lot the vocal production.
Mm-hmm.
But I think it still transmits energy, because I mean, with your line of thinking, the way that the animal vocalizes, it transmits information about the metabolic state of the animal, about the, right? So it's still valuable information in a social setting, right?
Yeah. So for rats, actually, I'm not sure, because I wouldn't expect that they use vocalization to communicate at a distance. They probably only use vocalization when they are close enough to each other, because their sound are very high pitched. They're like ultrasonic sounds, and those kind of sounds, I don't know, maybe they don't propagate very far.
I think it's just a matter of scale. I think it's still, I mean, it seems to me like it's a similar story in contact calls for some of these ultrasonic calls. Maybe the spatial range is smaller, mm-hmm. But yeah, they approach these calls, they emit these calls when they get separated and such. I think it is a similar story, and I see a huge correlation with locomotion as well. So,
I have a question.
Sorry, maybe just a comment. Sorry, just before going to just a comment. I think about the Diego's question, because there is the Morgan data sets. So there is a data set of marmosets, at least when they are infants, that there is a relationship between locomotion and vocalization.
Yes. Mm-hmm.
So I think it's pretty much goes in the Diego's idea that there is a kind of trade-off also. If the infant moves a lot, they vocalize kind of less, kind of.
Yeah.
So
yeah. And they always stop to produce Phee calls.
Mm-hmm.
Yeah.
But I think that's the opposite that it's observed in rats, right, Diego? That you see more vocalizations as the animals move, correct?
Yeah, that's a complete opposite. So
Yeah.
in an adult rat in a social setting, with some space to move, they will synchronize their vocalizations with movement a lot.
Oh, interesting.
So in a similar question, so I guess Diego is asking about arousal, right? So how does arousal affect this thing? And that sends me back, and I'll Sergio, I promise that I'll let you ask your question afterwards. Sends me back to the experiments that you did with the helium chamber. And whether if the changes that you're seeing is due to the changes in the auditory feedback and arousal in the animals instead of simply energy expenditure.
So it is possible, but for this case, we mostly focus on the respiration. So it's more about like the timing change. It is possible through auditory feedback, but so I'm not sure how auditory feedback act in this process as they're in heliox. And previous studies are mostly focusing on the spectral change by the heliox. Like they shift the pitch higher, right. They shift the amplified frequency band higher, and so that may have some effect on the auditory feedback, but it's mostly about like if they shift that resonance frequency, usually these animals, or at least humans, would also shift the pitch up to kind of match. But we observed in marmosets, actually, we don't really see such kind of match.
Right. So, but I guess my question is more related to the because you see that in other animals that are placed when you change, all of a sudden you change the auditory feedback, they change their vocal behavior in the sense that they are not sure what's happening and they don't quite understand what's going on. And I wonder if that has an effect on the changes that you're observing.
So it's very hard to tell to answer this question. But if it's something very surprising to them, I think we should expect that we can see some kind of adaptation later on. But I don't think I observe those kind of adaptation like in the beginning. They have more like disordered action because of this kind of effect, and then later on maybe it's reorganized into something more regular. But those kind of things I didn't observe, so I'm not sure. Yeah.
Sergio, I promise that I won't interrupt you this time.
Okay, thanks a lot for your talk. Can you hear me?
Yes.
My question is about the evolution history of the Phee call. This is very special to the marmosets because they seems to be absent in the capuchin monkeys. Although the capuchins they do how do you say in English assoviar, pfeifen, assoviar.
Whistle.
Whistle. Well, capuchins they do a lot to kind of whistles like.
Oh, interesting.
And this is not a Phee call. So my question is, do you think that first, is the Phee call only appears only in marmosets? If it is like this, do you think that then it was present in the common monkey between the New World and the Old World monkeys 25 million years ago? Or do you think it is something that the Capuchins invented?
So, so I have two angles to answer this. So one thing is that, so I'm not very familiar with other non-human primate species except marmosets. So that's my only experience. So I cannot speak too much of it. But this marmoset larynx has a structure. Let's see if I can find. Where's my mouse? I didn't include in my slides, but these this marmoset larynx. If you look at the anatomy of the vocal folds, you see that on top of this vocal fold there is a thin layer, like a membrane structure, and this kind of structure actually helps these marmosets to produce very high frequency calls because it can vibrate super fast. And so this kind of structure were also observed in some other non-human primates. And but there are very few studies about it, but people believe that it is like through evolution that they have this differentiated membrane structure that can produce this kind of high pitch and also very loud sound and very energy efficiently. And for the whistles, I think these whistles are produced by kind of different mechanisms. It's more like playing the flute.
Yes.
Right? So in that sense, I think it's the change. It's mostly by probably the vocal tract instead of the vocal folds, instead of the source, but by the tract. So maybe my guess, it's just my speculation, is that maybe these capuchins are more advanced in controlling their vocal apparatus because marmosets has very limited control of their upper vocal tract. Maybe these capuchins are like smarter. They can control also very well their upper vocal tract and produce this kind of whistles. So that.
Well, I mean, of course, I'm not an expert, but I have been working a long time with macaque monkeys, that's decades, and then I started working with the capuchin monkeys here. And the first thing I realized is that they have some commonalities of I don't know, some how to say, syllabs. But the marmosets are very different. And this is so amazing because, in fact, marmosets are also very different in other aspects, like having claws and things like this, and that they are so small. And I thought that this is really, really interesting. Thanks a lot.
Mm-hmm.
Anybody else? There's
yeah. So there is some questions I think in the chat, I guess.
Yes.
Yeah. Sorry, I have a question. That's fine. I was just waiting to. So one thing that I was wondering that in your hypothesis, so you are assuming that when the marmosets are become older, actually the coupling between the heart rate and the vocalization becomes stronger, actually.
Yes.
That's funny because I was always expecting that maybe to the opposite, because I saw that maybe when they are younger, the coupling is stronger, and then become more older, actually the coupling become more looser, and then let's say they become more cognitive control kind of thing. But that's not necessarily the case. That's interesting.
Yeah, but it's like 2-month-old marmoset is still considered very young. So maybe there's some other change after.
I see. Yeah. I see. So yes. Okay. I see. Yeah. In adults could be quite different. That's you mean.
Right. Yeah.
Okay. And maybe also one thing that it was not totally clear to me is that the patterns that you found in the resting states brain. Right? I didn't understand exactly. So do you think that those are those patterns are actually correlated to some specific phase of the arousal, the heart rates, or?
So I took a look. Actually, it's very complex. If you just look at one pixel, for example, and pick one pixel, pick heart rate, and you compare them, you see that at some moments their patterns are very similar. With usually I think the brain leading the heart rate, and also it's usually at some slow oscillations. But at some point, some of them may synchronize, and at some point maybe this heart rate will lead. So it's very hard to tell exactly what's happening there. I think it would be very nice to be able to take a closer look and have some ideas about what is causing this kind of different delays, and what's the contribution of frequency on that kind of delays. And also different pixels, like different brain areas, also have different phase difference with this heart rate. So yeah, so I think that's very interesting.
Great, thank you.
Perfect. You see, there's a couple questions here from our students. I'm asking Anna whether if she wants to ask directly, but I can translate. It's on the chat actually. Do you think, she's asking, do you think that when the marmoset is older, it chooses better the vocalization it will produce because it's now responsible for its own search for food in terms of energy supply?
What? I'm not understanding exactly this question. So
I think her question might be related to the evolution of the mechanism itself. Meaning, for instance, this is because there's a higher energy demand and they no longer it's responsible for its own search for food. Whether if there would be evolutionary pressure to actually select for a better selection of calls and not spend too much energy in it.
I see. So far what we understand are still very simplified, and it's based on the experimental setups, and which is not similar to what is going on in the nature. And in nature, the environment is more complex, and also the food resource would affect the behavior for sure. So I don't know exactly what the answer would be. It's basically we take this variable, which is just distance, to elicit different types of calls. But maybe this food resource would also contribute. And also, actually, the relationship between these marmosets who are calling to each other also matters a lot. If they are family members, they use certain type of calls, and if they're strangers, they probably won't communicate in the same way. And so I believe in reality the story will be much more complex. And in other species, for example, in fish, and people observe different foraging patterns of the fish, for example. It depends a lot on how rich in the environment the food resources are. And if the food resource is very abundant, and the fish would just randomly move. And then by chance, they'll see something they can eat. But if it's very sparse, for example, the food resource, the fish would take this thing called the levee flight that they take like long distance travels, like, and that is considered to be more energy efficient. So I believe for the marmosets, when they use their vocalizations, maybe they also have similar kind of adjustment based on just the environment. But I don't know what exactly that would be like.
Right. I actually have a question. It's more like a speculation because I'm not sure if you have ever done experiments. So if you think in a true communication setting in which you have the marmoset vocalizing your experimental subject, have you ever provide them with some vocalization feedback to see how that affects your observations about, you know, heart rates and how it's linked to vocalizations?
So I did some experiments to play back calls at different frequencies. So it depends a lot actually on the marmosets. So for some marmosets, actually they react a lot, like their heart rate reacts very strongly to the playback. But for some, they probably don't care as much. So so that's just like a very preliminary thing. I don't know if I can draw any conclusions, but I think it depends. So is Jeremy's paper also having that part of?
So I think so. Yeah. So Izzy is talking about, there is a paper by Jeremy Bourgeon, and he did a playback studies to see the heart rate change. But exactly as Izzy change, actually it's very complicated because it seems to depend on how the baseline heart rate of the animal. So if, for example, the baseline heart rate of the animal is high, actually, even if you do a playback, it doesn't seem to change much, you know. And if it's too low, also that's the case. So if it's in the kind of in the middle range, it seems to have actually higher effects. So actually, the effect is not like I would say 1-to-1. It depends a lot on the baseline heart rate. You know? So,
And the arousal state of the animal, right?
Exactly.
Yeah.
Yeah.
Excellent. Anybody else? Is there any more questions? Let me check on YouTube. No, it seems quiet over there.
I have a short question.
All right, Sergio, please go ahead.
Curiosity question. Some humans, it's very common that the parents change the pitch when talking to the infant. And I have heard, perhaps I'm wrong, that the macaque monkeys also do it, this kind of behavior. Marmosets also do it?
Maybe Daniel should answer this question. Isn't that the undergrads?
Yeah. It seems that actually that happens, but the funny enough, it seems to be mostly the father. The mother seems to don't care much, but actually the father seems to change the vocalization. But for marmosets, it kind of makes sense because except the first few weeks, most of the time actually the father takes care, and the males take care of the infants. And it's interesting that in marmoset's case, it seems that the father's call change more than the mother, and actually it's changes actually the acoustic characteristic of the fecals. That's one thing.
And this can be explained in terms of evolution, or do you think that there's a clearly social component?
I'm not sure. What we know, that the only thing similar that we know is that actually in adults, in humans, funny enough, it is something similar also. Actually, the father's call actually change more actually to when you compare to the mother. At least there is some evidence that that's the case. You know, so maybe there's some evolutionary reason for this, but I'm not 100% sure. Yeah, that's.
I think in finches you'll do the males do change their songs when they have youngs. I dubbed it fatteries because in humans it's very well known, right? Mutteries, the changing in especially vowels. The contours become more well defined, and that seems to facilitate vocal learning.
Yeah.
But there's also a lot of change, a lot of hormonal changes that are happening.
Exactly.
So we are not quite sure whether one thing is, if there has any meaningful changes there that will actually affect at least in finches. What I know is like the changes are very subtle, and it's not quite clear whether it modifies learning at all. It's very minute, the effect.
Yeah, I think in the Marmosets case also it's not like a huge, right? It's true, it's a clear Phee call, it just change a little bit to the acoustic pattern. But as you said, there is definitely change in testosterone, and that's known like for the male. So it's difficult to separate if it's a learning or if it's a testosterone effect. I agree.
Fantastic. If anybody has any more questions? If not, I would like to thank you, UC, so much for speaking to us. It was a fantastic talk. We all learned a lot, and we really appreciate your time to speak to us.
Thank you.
Thanks a lot. Thank you.
Thank you.
Thank you.
Thank you, UC. We'll see each other on next Friday. Thanks again, and you all have a good weekend.